Am I in the front of the slide? I will need to read now. That's okay? Yeah? Okay. So, okay, may I? So, hi again. I'm sorry that I'm here once again to present the third talk of this symposium. I'm going to present the talk title, Current Status of Phylogenetic Studies in the Genus Paspalum. This talk was prepared by Dr. Gabriel Hugo from Argentina. And this talk will include the results of several studies that were published by Hugo and his co-workers, especially people from Uruguay, Argentina, and Brazil. So here he listed Madalena Vaio, Marisa, Bonasora, Pablo Speranza, and I am listed here, but I'm only here to present his talk. Unfortunately, Gabriel couldn't come to attend this event, so I am kindly presenting for him, okay? So I apologize in advance because I will need to read most of the talk, okay? So the genus Paspalum, comprises about 350 species, and most of the species are confined to the neotropics. And the species of Paspalum are main elements of the biodiversity in South American grasslands, and they are seriously threatened by agricultural and forestry expansion. And also, several species of Paspalum are available as forage, especially in natural grasslands. We can exemplify this with the pampas in Brazil or the paramos, etc. And some of them are used as turfgrass. Well, the species of Paspalum are recognizable because of their plano convex spikelet. So if you look here, you see that the spikelet has one side flat and the other side is curved, so it makes the spikelet plano convex. And these plano convex spikelets here are arranged in unilateral racemes. And these racemes are arranged in several types of inflorescences. So inflorescences of Paspalum includes from one to several racemes. So it may be a single raceme, a double raceme making a V-shaped inflorescence, or three or more racemes that are distributed along the main axis of the inflorescences. In some cases, they are distributed in a flat fashion, or maybe they are arranged around the main axis, making a more three-dimensional structure. So some Paspalum are really beautiful, like the subgenus Serratia. Well, of course, Paspalum belongs to subfamily Panicoideae because it's in this talk. But within Panicoideae, we see here that Paspalum belongs to tribe Paspali, subtribe Paspalini. And within subtribe Paspalini, Paspalum is part of a clade that includes the genera Osvaldoa, Achia, Antenantiopsis, and Hopia. But there are no obvious morphological features that unite them. So once again, a lot of convergency. Well, between 2002 and 2009, several partial phylogenies were published based either on morphology or in molecular markers. So the first, more or less, comprehensive phylogenetic studies in Paspalum was this one here, published by Hugo and co-workers in 2010. So they carry out an analysis including 71 species, and they use 4 different plastid markers. And the genus here was recognized as paraphyletic because some smaller genera were recovered, nested within it. And also, we can see here the small lineage here. Paspalum winequivalvi was recovered outside the clade of Paspalum in plastid data. And here we see the small genus Tresiopsis within Paspalum that later in 2012 was combined in Paspalum to make Paspalum monophyletic. Also, the 2 main clades were recovered here in the Paspalum clade, and we can see that the backbone of the tree is poorly resolved, and because of several cases of allopolyploidy reported that could obscure the phylogenetic pattern, they also analyzed a submatrix including only diploids. So Scataglini and co-workers, they published in 2014 another phylogenetic study focused on Paspalum, in which they analyzed 131 terminals of Paspalum in related genera, and they used both plastid and nuclear DNA. And again, the plastid phylogeny yielded a poorly resolved phylogeny, and ITS marker provided a more solved topology. Here again, Paspalum in equivalve as well as Paspalum microstachyon, they were recovered outside the Paspalum clade here close to Antanachiopsis, Achia, and Osvaldoa in plastid DNA, but the ETS tree showed this clade here within the Paspalum clade. So an incongruence between plastid and nuclear data. And again, the small genera recovered within Paspalum, so Trasiopsis, once again here within it, already combined at that time. And also Spheneria, a very curious genus with clavate spikelets, and Rhematocloa, also within Paspalum. So both were combined to make Paspalum monophyletic. Further studies but done by Hugo and co-workers, unpublished data, involved 156 Paspalum species using the same 4 plastid DNA markers, and also generated poorly poorly resolved trees. I can't speak too fast. It's like ons. So again, poorly resolved. And here, once again, plastid markers showing Paspalum in equivalve outside Paspalum. So in the last years, plastome sequences of Paspalum became available. In the study of Burke and co-workers published in 2018, 11 plastomes of Paspalum were produced, and we can see here the small clade here that Paspalum was recovered as monophyletic, but notice Paspalum in equivalve here as sister to Paspalum vaginatum, and these 2 guys here as sister to the rest of the clades, so Paspalum monophyletic, including Paspalum in equivalve. So probably only a lack of informative data from previous studies. And an ongoing analysis of plastome sequences, so far comprising about 50 species, including the published sequences of Burke, produced a phylogeny consistent with the previous studies. And here we can see that Paspalum in equivalve, once again, recovered within Paspalum and close to Paspalum vaginatum, and the basalmost species here, Paspalum conjugatum. And it suggests us that the stoloniferous condition may be ancestral in Paspalum. Maybe it's a plesiomorphic condition. Well, it has been recurrently reported in Paspalum the occurrence of hybridization and polyploidy events associated with apomixis reproduction. For those who do not know what apomixis is, it's when the plant produces seeds or embryos without fertilization. So without the fusion of gametes, they produce viable seeds. So it's clonal reproduction. And this puzzles both the phylogenetic pattern because of the occurrence of reticulation and concerns related to the species concept, which throw doubts about the biological nature of the entities used as terminals in the phylogenetic analysis. So the following examples will illustrate some of these problems. So in the current taxonomic literature, 2 different biological entities are considered under the name Paspalum minus here. The typical one here is a pentaploid apomixis grass inhabiting warm regions of the American continent from Mexico to Paraguay. And the entity described here as Paspalum baratoi, here in the synonymy of Paspalum minus, is a diploid sexual restricted to cooler areas in southern Brazil. Well, a further diploid sexual species from southern Chile was described by Catanzaro and co-workers in 2015, and the scarce material available in herbaria had been also identified as Paspalum minus. So if we look here at this tree presented in the protologue, we note that the Paspalum minus was recovered here as sister to this clade, including Paspalum conduplicatum, Pumilum chilense, Berretoi, and Numularium. So here we see that the Paspalum minus complex actually includes 3 biological entities. So if we consider them as one species, we would neglect 2 sexual species, 2 sexual diploid species, and it's not good for conservation purposes. Perichon, in her unpublished dissertation, was successful in using low-copy nuclear genes to discriminate the phylogenetic relationships of each allele of 2 accessions of the pentaploid Paspalum minus. So here we see in this tree, obviously the chips are not here because this work was not published. So we see here that this red bars here represent the alleles of one of the accessions, so here, 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 and here, and the blue bars represent the alleles of the accession 2. So we see that a single taxa is placed in 5 different clades because it possesses 5 different genomes. One entity, one biological entity, so it's a chimera, basically. But it's an accepted species because it's apomictic and it maintains itself in nature, reproducing, producing viable seeds, and maintaining large populations in a large distribution. Well, the most exhaustively studied group of Paspalum is probably that of Paspalum dilatatum and related species. The forage value of this allopolyploid species and the difficulty in propagating it efficiently has long motivated cytogenetic studies for breeding purposes. Speranza in 2009 analyzed evolutionary patterns within this group using microsatellite markers, and his results, together with the previous cytogenetic evidence, led to a new classification of the dilatata group that was published by Virginia Russell and co-workers in 2022. The species of the dilatata group are the outcome of allopolyploidization events. Sorry, I lost myself here. Oh, so they are the result of allopolyploidization here, and this allopolyploidization involves 2 different genomes, genomes I and genomes J, and the crossings here produce these allopolyploids here, the dilatata group. So the Paspalum urvale, Dasypleurum, plurinervi, Poaceae, flavescens, and dilatatum, of course, the ones here in this tree, okay? And also, there are another allopolyploids from crossing between I and J genomes that are in the so-called Virgata groups here. Those in blue, Paspalum conspersum, Virgatum, Hergnelli, and Jurgensi. This is very crazy. Please don't freak out. I'll try to explain. I'll make my best. So this is those are the relationships among the entities of the dilatata group, and I will try to summarize it. 13 minutes, okay. So please take a look at here. This is Paspalum dilatatum, the subspecies dilatatum. This is the typical one. And we notice this is a pentaploid here, so genomes I, J, and X, okay? So look here that we have several entities of hybrid origin. So starting here at the top right corner, we see that a hybrid was formed between the typical dilatatum and an unknown mother, and this crossing here between this pentaploid and this taxa here generated an hexaploid that was called the biotype Torres of Paspalum dilatatum. Also, we see here that an hybridization event generated this clone 2 by crossing the typical Paspalum dilatatum with the Paspalum dilatatum biotype Virasoro. So this tetraploid here and this pentaploid here generated this pentaploid here. In some of these crossings, you will see that a regular meiosis and in others, incomplete meiosis. So unreduced gametes. Oh, sorry, I have to come back. I only explained the beginning. Oh, go back, please. Yes. So here we have several hybrids. So this biotype here, Uruguayana, was generated by crossing between the typical dilatatum and an unknown father probably from the Quadrifaria group. And so this pentaploid here with this guy here generated an hexaploid. And here this clone 59B was generated by crossing by the typical dilatatum and donor from the Quadrifaria group as the father and generated an heptaploid. And here the biotype Shiru was generated from crossing clone 2 with an old father from the Quadrifaria group, generated an hexaploid. And here we have a species that was already circumscribed, Paspalum L, a tetraploid from crossing clone 2 and an unknown mother. And also, there were other species in the dilatata complex, Paspalum urvile, Dasypleurum, and two biotypes Vicaria and subspecies Flavescens. So, this is a summary. Here we see that this biotype here, this subspecies, and these two guys here, they are all sexual tetraploids. Okay? And all the other entities here are polyploids, apomictic ones, in several levels of ploidy. So, all these entities here used to be called as Paspalum dilatatum sensulatum. What was done with this group? Okay, so Russell et al., they did this. They kept Paspalum pauciciliatum as a tetraploid apomictic species. They kept Paspalum urvile, Dasypleurum, and also they elevated some of these biotypes or subspecies to the species level. So the sexual ones here, the biotype Vacaria was transferred to Paspalum vacarianum, the subspecies Flavescens to Paspalum flavescens, and the biotype Virasoro to Paspalum plurinervi. And the apomictic ones, some of them were regarded as the Paspalum dilatatum with some varieties. And this biotype here, apomictic one, hexaploid, was described as Paspalum torrense. So we can see this very complex evolutionary history depicted in one species complex. Okay, so let's move on. Finally, Paspalum stellatum comprises a complex that includes a diploid cytotype with 20 chromosomes and a set of polyploids having 32, 44, 46, 48, 52, 56, and 60 chromosomes. So the cytotype of 32 chromosomes is the most commonly found. The discovery of Paspalum Chesley related diploid species with 12 chromosomes threw light on the origin of such irregular polyploids. In fact, Bonasora and co-workers in 2015 proposed that the 32 cytotype here is an allopolyploid resulting from hybridization event between the diploid Paspalum stellatum with 20 chromosomes and Paspalum Chesley with 12 chromosomes, and it generated the 32 cytotypes that behaves as a sexual amphidiploid. Successive backbreeding events partially explain the formation of higher polyploids, which are maintained in nature thanks to apomitic reproduction, so all apomitic populations here. Discriminant analysis of morphological data show that morphologically, all the polyploids resembles Paspalum stellatum, so they have been currently classified under this name. Nevertheless, the taxonomic status of these entities is now being reappraised, taking into account the hybrid origin of all polyploid cytotypes. So to conclude here, we can outline some final considerations. The genus Paspalum is monophyletic now because those smaller genera were combined in Paspalum. And the infergenetic taxonomy of Paspalum needs to be reappraised because the previously recognized subgenera were not fully supported. Analysis of whole plastid genomes allow us to obtain well-resolved phylogenies, but species sampling is critical at a current stage, so many efforts should be made in this regard, including the provision of financial support and the reinforcement of collaborative work. And we must not lose sight of the fact that phylogenetic hypothesis obtained from plastid DNA only reflects maternal relationships. And in a group like Paspalum, that hybridization and polyploidizations are very frequent, other analytical tools would be necessary for a complete understanding of the phylogenetic relationships and the processes that explain them. And the use of low-copy nuclear genes seems promising, as well as the separate analysis of diploid materials to obtain the backbone phylogeny for the noise, free of the noise caused by allopolyploidy. So I would like to thank, on behalf of Dr. Rua. Thank you. Hands up.
Thank you very much. Questions?
I can try to answer.
We are plenty of time.
I think we burn all neurons.
I see that, but I think that it's a problem in many areas of science. But this problem with the funding. So what we can do?
What we can do? Write projects, submit them, and wait.
But in that case of Poaceae, how can you?
We have several options. In our countries, usually we have the financial agencies connected to research programs, and usually we apply submitting projects to get money. We have private initiative, but in Brazil it's not common. And we have the difference between countries. So people from the United States usually say, oh, it's very, very easy to sequence, to do NGS, but for us, yes, it's very hard to buy paper to mount herbarium specimens. So sometimes it's not that easy, but we try. Yeah. Collaboration is the key.
Hello, you showed us a case about the Paspalum L. Please show the slide again, would you?
Oh, can you please put the presentation back? One moment.
Yeah, you mean that?
Oh.
One taxon, and in fact, it belongs to 5. Yeah.
Yeah.
That means, okay, the homoplace happened 5 times in the different evolution branches.
Not necessarily. It means that this entity possesses several parts of different genomes of different species. So when you sequence them, you have paralogs. And some paralogs will fall with close to the parent 1, and the other paralogs close to the parent 2, the other paralog close to the parent 3, and the other paralog close to the parent 4, and the other paralog close to the parent 5. That's it. So it's really a chimera, really a chimera. And we all are. Plants are chimeras, but plants pass through diploidization events. They produce polyploids, and through time they become diploid. They become diploid by losing random parts of both genomes. So now we have angiosperms as diploids, but they have a polyploid origin. It's comprovated by the studies of Doyle and Doyle. Yeah. Okay. It's a very complex part of the evolution that we usually don't look at.
Okay, I think I have to discuss further with you later.
Okay.
Okay.
That's it.
Thank you very much.
Thank you.